Hello students, today we discuss the continuation of sexual reproduction in flowering plants. Last class we discussed what is the sexual reproductive part in a angiospermic plant, that is a flower under. In this flower, due to growth and development, that means due to maturation on these floral parts under, that means premature part we call as primordium. With the help of hormonal actions, this primordium it develops all four oral parts and on the essential floral parts are androecium as well as gynoecium. Androecium that represent as this one, that is a male part, it is called as male reproductive organ. Similarly, female reproductive organ, that is gynoecium. Female reproductive, if it is both are present in one flower, that is we call as a complete flower, that is a monoecious flower, that is a bisexual flower. Then in this androecium, what are the parts present in androecium? A group of stamens. A group of stamens collectively we call as this androecium and all. In this, a group of stamens present. Each and every stamen. So each and every stamen, one long, that slender stalk is present and all. What is the name of long, slender stalk and all? This is only. This is we called as filament. This is we called as anther. See here, the long slender stalk we called as filament and all. This is the ventral view, this is the dorsal view. In the dorsal view, here it is connected with the connective. It is connected with the connective tissue and all. There is a vascular tissue and all. So, a long slender stalk which is present in stamen, what it represents, that is regarded as a filament and all. In a filament, what is present in a terminal part and all? The terminal part of filament, it is present short bilobed anther is present and all. Once again, short bilobed anther is present. Why? Because related to mismatching, that is important. Long slender stalk, hyphen, filament, long diathecous, that means bilobed hyphen anther, short bilobed anther, long bilobed anther, like this one. Here, one among this one, short bilobed anther is present and all. Another very important related to, in this long slender like stalk we call as filament. Just as I said, in a terminal it is present bilobed anther, it is present and all. Whereas in axial part, that pre-axial part, that means the basal part of the filament where it must attach and all. The axial, that means the pre-axial part attaches to thalamus or sometimes maybe corolla, that means a petal. So that is a pre-axial part or the basal part of filament. It is a NEET question. 2016 they asked in that one. So what is the pre-axial part of filament? It must be attached. So where axial filament it must attaches. It must attaches on the thalamus or petals and all. So they must identify any one among the correct statement and all. They give it attaches on that towards the stem, it attaches towards the petals, it attaches towards the thalamus, or none of this. Variously they ask and all. So you must remember the basal or pre-axial part of filament attaches either thalamus or petals. Some of the rare cases it attaches to the style also, but more frequently it attaches to the either thalamus or petal cell. Then the terminal end, that is just now we said, that is we call as anther, that is more commonly bilobed anther and all. So these stamens are different flowers, different number, different sizes of the anthers are present and all. But a typical angiosperms, either dicots or monocots and all, most common type of anther will be the bilobed and all. It is present two anthers, that means two lobes and all. This is one lobe, this is another one. Both we call as bilobed one. Two are present, that's why it's called as bilobed one. It is connected with the connect one. One on this side, it must be visible very clearly and all. Bilobed one. So each lobe here, each lobe it contains, there is a two theca. Each lobe it contains two theca. If you observe three-dimensional section of this one here, this is one theca. This is the two thecas and all. Two thecas are present inside this one, that is the microspores. This is dehiscence. Everything will come in further and out. So in three-dimensional, if you observe, this is one theca, 2, 3, 4. So each and every it will be present. One lobe is present, two theca, like that. How many number of thecas are present? 4 thecas are present and all. In between two lobes, there is a thin membrane. There is a thin membranous spot is present and all. Between these two, it will be present thin membrane it will be present and all. This is the thin membranous, this is otherwise recorded as stomium. Stomium recorded as. So this is between two theca. So this is one, this is another one. Like this, so 4 it is present. Between this one, here it is present connective. That means vascular tissue connected between the two theca. This is one particular lobe. One lobe has present two theca. Between two lobes, it has present very thin membrane bounded walls are present. That is we call as stomium. Wherever stomium, that respectable part ready to dehiscence of the whatever the male gametes or whatever the male gametophyte, that means microsporangium, which dehiscence towards this particular manner and all. Between two theca, that connective tissue is present. This is the connective tissue and all. Then in this one, this also we represent of this one. Each and every it will be formed into the tetragonia. Tetragonia means this is one, this is another one, this is another one, like this. That 4 microsporangia will be formed and all. 4 microsporangia. Each and every we call as microsporangium. Microsporangium, like this 4 are present. Each and every microsporangia, otherwise we call as pollen sacs and all. Pollen sacs. Microsporangia. So this is one microsporangia. Inside this one, whatever lined one, this is only we call as microsporangia. This is outer membrane. That is a common outer membrane throughout all microsporangia. The spherical part only we call as microsporangia. The spherical part only we call as microsporangium. Then each and every microsporangium, how it must be formation of the particular cells and all. In this one, in this Microsporangia, in tetragonia outer, it is formed into the exothecium, otherwise we called as epidermis. It is present throughout all the membranes and all. The inner archisporium it will be present. The inner lining archisporium is present in archisporium due to periclinal cell division with the help of periclinal division. With the help of periclinal division, this archisporium differentiate into the outer, that primary parietal cells, then inner primary sporogenous cells and all. Parietal and sporogenous. So outer throughout this one we call as epidermis. Below the epidermis, whatever the layer, this is we called as archisporium. This archisporium again due to mitotic cell division periclinally it is segregated into the inner sporogenous, the outer parietal cells and all. The parietal cell due to mitotic cell division again it differentiate, again it formation of the membranous, wall membranous cells and all. It forms wall membranous layers are formed. What are these one? Wall membranous layers, that is endothecium as well as middle layer, that is 1 to 3 layers, then tapetum and all. The sporogenous cells collectively to form as the central point of sporogenous one. It forms into the sporogenous tissue and all. It forms sporogenous tissue. Then this sporogenous tissue, that means a group of cells, it differentiate or it converted as this one sporogenous mother cell. Sporogenous mother cells. So exam point, that is important. What is the ploidy of sporogenous mother cell? Otherwise we call it as pollen mother cell. What is the full form of? That is one pollen mother cell. Pollen mother cell. So from where pollen mother cells are formed? Pollen mother cells are formed inner primary, that is the sporogenous cells which undergoes the formation of that specific sporogenous mother cell, otherwise we call it as pollen mother cell. What is the ploidy of pollen mother cell? That is the diploid and all. This particular pollen mother cell, it differentiate into the, it will be through meiosis cell division in formation of microspores or pollen grains and all. So that is important. That is the process of formation of microspores from microspore mother cell. Once again, the process, the formation of microspores from microspore mother cell or pollen mother cell. That is we call as microsporogenesis. This particular process we call as microsporogenesis. So this is also NEET question. How? So what type of cell division requires microsporogenesis? Similarly, megasporogenesis. Megasporogenesis related to formation of female. Microsporogenesis formation of male. So they ask microsporogenesis undergoes what type of cell division, or which type of cell division required to formation of microspores from pollen mother cell. They must give option either meiosis, either mitosis, both meiosis, mitosis. Then you write the correct answer is the meiosis cell division. So microsporogenesis, it undergoes or it involves meiosis cell division. That is most important. Meiosis means already the particular mother cell related to diploid condition. That is innermost one. Then what is the parietal one? So what is present between microsporangium as well as epidermal? Between these one. So between these one, what are the cell layers? What are the cell layers which are present in that parietal cell? Already we said. What among these one? Endothecium. Then one or two layers of this one. There is middle layers. The innermost membrane. The innermost membrane, that is nothing but tapetum. It is present now. If you observe in this particular view, if you expand it under, then outer it is present epidermal layer. Outer it is epidermal layer, then inner the fibrous endothecium is present and all. Inner fibrous one. Later 1 to 3 layers of middle layers. 1 to 3 layers of the middle layers. These are middle layers of this one. Middle layers, 1 to 3 it is present. But the innermost membrane, little bit elongated cell. Apart from this one, multinucleated due to endomitosis. It may be forms into the polyploidy and all. Some of these one elongated, innermost elongated cells related to multinucleated polyploidy. It is present and all. Related to this one, outermost one, that is the epidermis. So that sequence of order. So whatever I drawn here, if we expand like this manner, it will be presented off. Then endothecium. Endothecium, it is covered with cellulose and all. That's why it is fibrous layer. It forms into the fibrous. It is a fibrous membrane. Whenever pericliner descends and all, then the fibrous layer act as a vital role, or very important, which type of the membrane act as vital role? Descents of male reproductive, that is male amplified, that means pollen grain, microspores are pollen grain, with the help of endothecium. Why? Because remaining all membranes before descents, it must be degenerate and all. It will be persist. At the time of drying only, it rupture and separation of this one. Then these are the middle layers. Middle layers, 1 to 3 layers. It is the parenchymatous cells are present. The innermost one, that is the tapetum. That is important. The first 3 layers. What is the important role of epidermis, endothecium, as well as tapetum and epidermis, endothecium, tape, that is related to middle layers. All these 3, it must be helpful for protection as well as descents of the pollen grains. It helpful mainly protection and descents of pollen grains. And descents of pollen, that is important. So what are the membranes which helpful protection and descents of the pollen grains? Epidermis, endothecium, middle layers. Some rare, minorly, that middle layers, it helpful the nourishment and all, not a vital role and all. Majorly protection and descents and all. Then the innermost. So exam point of view, it is very important. What is the outermost as well as innermost membranes present each and every microsporangium or pollen sac and all? They give option: epidermis, tapetum; endothecium, tapetum; middle layer, tapetum; or epidermis, middle layers. Like this, multiple options they give. According to question, you must remember what is the outermost layer, what is the innermost membrane. Outermost membrane is the epidermis. Innermost membrane is the tapetum. Whatever the layers, it may be, but outermost as well as innermost, that is important. But what is the vital role of tapetum? The vital role of tapetum, it acts as very important nourishment of the sporogenous cells, nourishment of the pollen mother cells, nourishment of the pollen grains and all. So, majorly helpful, the tapetum acts as a vital role for nourishment of that specific part of the pollen mother cell or microspores and all. So the tapetum cells act as a vital role. So nourishment of pollen mother cell or microspores and all. Apart from this, what type of nourishment? It helps for secretion of enzymes and also food material, as well as that enzyme is nothing but it will be produces callase. It produces callase enzyme and all. And also, one among the scientists explained the function of tapetum. The name of that scientist we called as Ubish and all. That's why that is Ubish bodies. That Ubish bodies, it produces sporopollenin. It is helpful for the formation of outer membrane of pollen grain. Outer membrane of pollen grain, that means exhale. That means exhale and all. So, Ubish scientists identified some lipid bodies are present, that is the Ubish bodies, which are secreted by the tapetum cells, and these tapetum cells, the particular chemical sporopollenin, further it helpful outermost membrane of the pollen grains and all. But it must be withstand extreme heat, extreme acid, extreme alkaline. At any extremity, it is also need question. What is the functioning of sporopollenin? Even due to presence of sporopollenin, it may not disintegrate, it may not degenerate, even extreme temperature, even extreme acidic nature, even extreme alkaline nature. That's why it must preservative one due to presence of sporopollenin and all. Then, apart from this one, pollen kit. Whenever the pollen grains are transferred to insects and all, then that is we call as pollen kit, and such type of pollen grains is presented to the pollen kit and all. Then this tapetum, according to their function and amoeboid or invasive, as well as secretory or glandular. So amoeboid tapetum cells, all cells fused and formation of plasmodium, and invasive, the adjacent of this pollen tetrad or pollen tetrad and all, the adjacent of the pollen microcells and all, that is pollen mother cells and all, adjacent of the invasive and formation of a plasmodium in between pollen tetrad, in between pollen tetrad for nourishing of these indirectly. So, to amoeboid cells, while growth and development of pollen grains, it degenerate, it must be enter innermost layer of this one. Here only that respectable of pollen mother cell, it forms into the pollen, that is the PMC. It undergoes into the meiosis, then it forms into the pollen tetrad. In between pollen tetrad, it enter and nourishing proper growth and development of pollen grains and all. But it must be in situ or parietal, without detaching from that particular cell. It secretes the substances and the nourishing of the pollen grains and all. And the nourishing the pollen grains and all. But it degenerates. So what type of plants normally it is present and all? Normally amoeboid, such type of tapetum cells, more probably we observe this amoeboid one, example, like typha, one among these one, like typha, as well as lily, then Aristolochia, like typha, lily, Aristolochia, such type of the plants, amoeboid, invasive tapetum cells are present, whereas secretory glandular cells, normally it is present symphoric corpus. Once again remember, amoeboid tapetum cells, normally it is present into the typha, lily, as well as Aristolochia or alisma, not Aristolochia, not Aristolochia, alisma, lily, typha, or as well as Radiscantia, such type of plants are present. It is present only symphoric corpus, such type of plants it is present, secretory and all. Even though whatever it may be present, after secretion, after nourishing, it must degenerate. As well as after maturation, pre-maturation only, all these epidermis as well as endothelium, middle layer, tapetum is present. After matured, after developing that particular pollen tetrad, pollen grains and all, then epidermis, middle layer, as well as tapetum cells, it will be degenerate. Only endothelium it is present. Then after drying of this one, it ruptured and periclinally it must be dehiscence and all. It must be dehiscence towards the periclinally, it must be dehiscence. Other than that particular dehiscence, that cells also it will be degenerate normal only. After maturation only, it will be degenerating, that particular part of this one. Then in that pollen mother cell, through meiosis, pollen tetrads are formed and all. This pollen tetrad, in what is the particular different shapes of the pollen tetrads normally observable? So after meiotic cell division, the pollen tetrad. So normal types of pollen tetrad. Pollen tetrad. So normally it is present into the tetrahedral. Tetrahedral, example, dicots and all. Or this tetrad it is present isobilateral. More commonly we observable into the monocots. So it is the most common. That is very important example I tell you. So which type of, what type of plants normally we observable? That is the tetrahedral shape of pollen tetrad and all. Then eruption will be the mostly in dicots and all. Then isobilateral it is observed with the monocots and all. Then whereas it is also observable linear as well as and T-shaped and all. It is also observed to the, that is the T-shaped, this is the linear. Apart from this one, this is the decussate. So all these tetrahedral, isobilateral, linear, T-shaped, decussate, all these 1, 2, 3, 4, 5, it is present into the Aristolochia elegans and all. All different shapes are present into the Aristolochia elegans. So Aristolochia elegans plant, all remaining parts like a T shape, like a decussate shape, like linear shape, also it is included to the tetrahedral as well as isobilateral. Every, all it is presented to the Aristolochia elegans and all. So that is related to tetra, that is the pollen tetra means after meiosis, all 4 microspores are functional and all, except cyperaceae. That is important. In cyperaceae, only 1 microspore is functional. In cyperaceae, normal remaining all other particular plants and the after meiotic cell is most important. meiotic cell division, all 4 microspores are functional except cyperacin. Cyperacin, rest of all other 4 functional microspores are present. Microspore nothing but pollen grains. All get together, it is present in the form of the tetrad and all. How it must be connected with each other with the help of callus. It must be connected. Suppose here to here, it is connected with the help of the callus. That means beta 1,3 glucan. That is nothing but beta 1,3 glucan. With the help of beta 1,3 glucan callus, that is very important. So callus membrane it covered with. They give options: pollen grain, pollen tetrad, Microsporangia, or any other. They must be given or ovum and all. So your option will be the pollen tetrad. So normally callus membrane it must be covered in that pollen tetrad and all. It is made up of the beta 1,3 glucan, that is specific chemical and all. But after maturation of this one, it is also degenerated. It is also disintegrate. Then to dispersal of the pollen grains and all. Then these are pollen tetrads are present. Some of these organisms, total microsporangia, it filled with the pollen grains and all. It filled with the that is microspores only. That is we call as collectively formation or collectively presence of pollen grains, microspores inside the microsporangium. We call as Pollinia. What we call as Pollinia. So like this. These are we called as Pollinia. Collectively, all pollen grains present inside the microsporangia pollen sac. Whatever pollen sac earlier we drawn and all, inside total, instead of pollen tetrad, all pollen grains get together, it is present and all. That is we call as Pollinia. Normally this Pollinia we observe to the orchid as well as Asclepias and milkweed, that is Calotropis, milkweed Calotropis and all. Whereas in that Calotropis, it is attached to the corpusculum as well as cadicle. These are microspores and all. Inside this one, inside whatever it may be, all are it is present into the microspores. All get together, microspores are present. But this is we called as translator mechanism. Normally we observe to the Calotropis. So corpusculum, cadicle and Pollinia. So normally orchids and Asclepias, Pollinia only is present, whereas the Calotropis like milkweed is present. Both the Pollinias and that is here it is connected with the corpusculum, then cadicle, corpusculum, cadicle, Pollinia get together we call as translator mechanism. That is important. Which type of the plants that translator mechanism normally observable. Then your option will be the Calotropis or commonly called as milkweeds and all. So after complete maturation, that means with the help of whatever tapetum cell callus. So whatever callus, with the help of callus, then separation of microspore from tetrad. With the help of callus, it must be separated. So after maturation, so after complete maturation, it is present. After complete maturation, it is present like this manner. it is a complete maturation. So TS of Anther, that is a matured one. These are pollen grains and all. Toward the stomium, it must be dehiscence and all. It must be dehiscence. That means in that one, endothecium act as a vital role. Even though after a degenerated remaining one, it must be persistent here. So, once or twice, you observe very carefully. So, what is the part which are present in stamen? What is that slender stalk part? Where pre-axial, that means the innermost part, that means the prominent part, the basal part of the particular filament where it attaches and all. So what is the typical particular type of the anther and all? That is the typical anther we call as bilobed and all. In that one, how many microsporangium are present? 4 microsporangium. It is present in corner side, 4 microsporangium. Each and microsporangium, what it develops? Each microsporangium, that means pollen cell, through it converts into some of these cells, sporogenous cells, it forms into the that is the pollen mother cells and through meiotic cell division, microspores or pollen grains are formed and all. So in this lesson, what we studied, microsporangium. Inside the microsporangium, microspore mother cell through meiotic cell division, microspores, that means the reproductive parts are formed and all. That is male gametophytes are formed and all. In this one, what are the outermost layer? That means epidermis, endothecium, middle layers. That is innermost one, tapetum. In that one, which one acts as a vital role for nourishment and tapetum cells and all. So, in that, after maturation, the endothecium acts as a vital role for dehiscence of this dispersal of this one particular pollen grains and all. Okay? Remaining to discuss in next class and all.